Molecular evolution. Antibodies diversity originally from germlines, the gene shuffling or gene recombination, and then somatic hypermutation make a further diversity, and eventually it make out the somatic antibody. Then what is the unit of hypermutation? B cells change its sequence by high rate point mutation. So the unit of hypermutation and selection is a cell. If you think about the size of a B cells, it's a little small size of a cell. Still, that's a 5 to 8 micrometer size. If it becomes plasma cell, that becomes 10 to 20 micrometer. So approximately speaking, human cell or mammalian cell is about 10 micrometer size. In comparison, yeast cell, which is also eukaryotic cell, which is almost a half the size of a human cell, if the length is half, the volume is 1/8, that's about 10% of the human cell. If we want to have a higher diversity, then maybe using smaller unit may be more helpful to get a higher diversity. How about the bacteria? Bacteria is about 1 micrometer size. Size-wise, 10% of a human cell, then volume-wise, it can be 1,000 times smaller. Then, if you use a bacteria as a unit of selection, maybe the diversity size can be much, much bigger. Here is very interesting model of the selection. So M13 bacteriophage is kind of a virus, and it has the DNA as a gene, and utilizing those genes, it can make a protein. And the size of this virus is 10% of a bacteria, about 0.1 micrometer. So if you utilize this the bacteriophage, the virus as the selection the unit, then the diversity number can be increased a lot. This is how we can make phage display. The first step is the DNA library preparation. This is the source of diversity, and also this DNA library even can be prepared by chemical synthesis. This library can be inserted in plasmid, circular form of DNA, and this plasmid can be inserted in bacteria, and this bacteria can be added with a helper phage. Then they can assemble together inside this bacteria. Now they can generate huge library of a phage display. Then they can select the proper target protein, and then through this displayed protein, and they can bind to this target protein. If they do not bind strongly, then they can be washed out, and only bound form can be selected, and then extract out this phage and analyze those genes. Then these genes can be reutilized and to provide the amplification of the cycle. So through this amplification cycle, now the most strongly binding phage can be selected by this phage display panning cycle. Now, to introduce the diversity into the DNA, there are several strategies. One approach is just a random mutation of the gene, or selected point can be focused, the diversity can be generated, or similar to the recombination of the genes, recombination method can be also utilized. Phage display method is very similar to evolution process. So evolution is composed of a diversity generation through mutation and selections. And those mutations is recorded in DNA. And the selection unit can be either individual or maybe cell or even virus. If you can change those selection unit into just the protein itself, now it's a cell-free. Now this whole system is DNA to protein. Then in this level, maybe we can call this as molecular evolution. mRNA display is an interesting model of a molecular evolution. So ribosome is the machinery to make a protein. The input information is mRNA, and output is a produced protein. By adding the antibiotic and genetic modification, we can block the release of mRNA and release of protein. Now there are one complex of genetic information and product together, and using this complex we can do screening. Then we can figure out the identity by analyzing those mRNA attached. So in this system, now the ribosome is the big chunk, and the size of ribosome is about 20 to 30 nanometer, but still this is much smaller than virus. So if you want to have a much higher diversity, even comparing to phage display, mRNA display can be one good example. Now, can you apply this idea to chemistry? Well, here, there's a selection and variation, and weakly functional molecule we wanted to evolve to the improved version. And maybe we can select those selection process. So mimicking evolution process. And so far, all the product was a naturally occurring peptide or protein. Can you change this into chemical synthesis? DNA encoded library is a perfect example. When we synthesize chemical through linker molecule, we can record that information into DNA. And when we modify the chemical structure with some reaction, and that history is also recorded in DNA, and next step also recorded in DNA, then this kind of a chemical can be synthesized in parallel to make millions of mixtures. Then this molecule can be screened through binding assays. Then very small amount of binding to this target can be isolated out. And those molecule amount itself is very small, but it carries this DNA information. DNA information can be easily amplified by PCR. By analyzing this DNA sequence, we can decode the structure of chemicals. Using this beautifully designed method, now it's possible to test millions of compounds in one set of screening.